Hey everybody, welcome back to another lecture video. In this chapter, we're going to take a look at what finally happens to the products of glycolysis and how the electron transport chain turns those products or intermediates into energy. So the citric acid cycle is a bunch of reactions, and I'm going to show you each of the steps in a moment. It basically, like it says here, just connects the intermediate acetyl-CoA from stage 1. So remember, we've got glycolysis, which is stage 1, in which a glucose comes out as 2 pyruvates. Okay, the glucose had, 6 carbons. The pyruvates each have 3 carbons. Now, in stage 2, the pyruvates react with our coenzyme A to make acetyl-CoA, which is a 2-carbon group. And when there was 2 pyruvates, we're going to have 2 of these acetyl-CoAs, and we released in that step 2 carbon dioxides. So our 6 carbons have now turned into 4 carbons, right? Because there's 2 of these acetyl-CoAs, and those have 2 carbons. The other carbon from each of those pyruvates got breathed out. And so now this intermediate, the acetyl-CoA, we're going to look at stage 3, which is the citric acid cycle. So this operates under aerobic conditions, meaning oxygen has to be around. We'll see where that oxygen comes in later in the mitochondria. Our 2-carbon acetyl group is going to be basically converted into carbon dioxides. So we're going to breathe out carbon dioxide. That's how we oxidize all of the sugar, all of the carbons from the sugar. Now, so we'll keep track of those CO2s. The coenzymes, NADH and FADH2, that we looked at in glycolysis, are also going to be produced here. The name comes from citrate, when we name this thing as the citric acid cycle. It's also called the tricarboxylic acid cycle. And when I learned it, it was called the Krebs cycle, named after probably the guy who first sort of elucidated this process. But now we call it the tricarboxylic acid cycle, the TCA cycle. Okay. So that's just a little bit about the name. And as I mentioned, we're going to be following the carbon dioxides that come off. And so in the next slide, I'm actually going to show you why this is a 6-carbon, why they say 6 carbons move through the citric acid cycle. Really, it's a 4-carbon molecule that we're going to regenerate at the end of this citric acid cycle, so that our 2 carbons in form of acetyl-CoA will add in to make a 6-carbon compound. And then as this goes through the citric acid cycle, we're going to be breaking down, or we're going to be losing carbon dioxides along the way. And so that's how we're going to go from 6 carbons back down to 4 carbons, ready to take in another round of acetyl-CoAs. It's important to remember that this whole process happens twice for every 1 sugar molecule. Remember, because there's 2 pyruvates, 2 acetyl-CoAs, and each time the citric acid cycle goes through 1 cycle, it's only working on 1 of those molecules. So we're going to count the CO2s, we're going to keep track of our electron carriers, and we're also going to note the fact that we're going to produce some ATP directly, just like we produced in glycolysis. So here's a better sort of view of the reactions, there's 8 reactions that actually oxidize the acetyl-CoA. And this acetyl-CoA can either be from pyruvate or from fatty acids, which we haven't looked at in this chapter. We'll take a look at in the next chapter. And so you'll see we're going to produce CO2 in 2 steps. Not every reaction is shown here. We're going to produce some high energy compounds, FADH2, NADH, and some GTP. This will go into the citric acid cycle to make more ATP. All right, and the reactions that are involved in the citric acid cycle are varied. There's condensation reactions, oxidations, reductions, hydrations, dehydrations, hydrolysis. You should be able to kind of guess what each of these would be doing. So just as a reminder where we are, so we are now looking at stage 3, where stage 1 would have been breaking down essentially our molecules so they're small enough to get into the cells. Stage 2, where we start to actually break these down into even smaller molecules, and some oxidation starts to happen, like in glycolysis, for example. We start to oxidize glucose a little bit while we break it down. And then stage 3 is going to be entering the citric acid cycle and producing the electron carriers for electron transport. So we're going to take a look at those reactions now. So we'll start with reaction 1. So we can see here acetyl-CoA is going to come in with our 4-carbon oxaloacetate and a water. And there's an enzyme here, citrate synthase. We're going to basically combine that acetyl portion onto the citrate. Coenzyme A, which we'll call now, what do we call this now? It's not acetyl-CoA, and it's not the coenzyme A. I'm forgetting what we call this guy, now that it has that thioester bond on it. I guess it's just coenzyme A. So what happens here is that we get a transfer essentially from hydrolyzing this thioester bond that gives us enough energy to form a new bond here. So then we get citrate. Citrate enters into the citric acid cycle. So in reaction 2, citrate rearranges into isocitrate. So that involves the enzyme aconitase. Aconitase also catalyzes the dehydration of citrate to yield cis-aconitate, following by hydration. So in a 2-step process here, citrate essentially gets rearranged into isocitrate. Now this is all done in preparation for the next step, reaction 3, where we're going to undergo some oxidation. So oxidation here in this case is going to be done by isocitrate dehydrogenase, and we're going to have a decarboxylation here. Now the reason I'm saying that there's an oxidation happening is because this is going to be reduced to NADH, which means there has to be something oxidized. But in addition to that, we can see the decarboxylation yields a carbon dioxide molecule. So already one of the 2 carbons essentially that we brought in is getting breathed out. Now you'll notice that it isn't one of the 2 literally that came in on the new sugar. They get recycled. So one of the carbons that was here from probably a previous cycle is getting breathed out, and then eventually these guys will become part of this structure, and we'll have a new set of these guys for the next round. So all the carbons eventually get breathed out. It's just not the exact ones that came in on this round of pyruvate. Okay, so then we get some electron carriers, we get some CO2 in this step. Reaction 4, so we have another decarboxylation and another round of reduction of NAD+. So we get some more electron carrier, we get some more carbon dioxide. I forgot to mention in the last one, our isocitrate was turned into alpha ketoglutarate. Now, alpha ketoglutarate in this case is going to be um, essentially oxidized into succinyl CoA. So now we have a new compound here. Oh, there was also this guy, this coenzyme A came in here too. So this gets reduced, this gets oxidized, and this guy gets decarboxylated. The energy of that allows these guys to connect, and so we end up with succinyl CoA. So you can see another carbon dioxide is breathed out. That's the important part here, and we're not even looking at mechanisms or anything. So all we're really focusing on is carbon dioxide was produced again. So now we've breathed out our second carbon. So all of the sugar essentially has been oxidized. The point now is to try to get succinyl CoA to turn back into oxaloacetate, which was the first molecule in our citric acid cycle that accepted the acetyl CoA, right? We want to regenerate this process. So succinyl CoA will now react and be essentially, well, what's the word? Converted, or I guess in this case we're going to lose our we're going to hydrolyze this coenzyme A bit. That's going to produce enough energy to actually add a phosphate to a GDP, a guanidine diphosphate, and turn that into a GTP, which is essentially the same as an ATP. These are high energy molecules. So that step generates an ATP directly and also gives us a 4-carbon succinate compound, which then in reaction 6, we're going to take succinate, and here we're going to use it to oxidize fumarate. So fumarate here gets, succinate gets oxidized and FADH gets reduced. I think I was calling this guy fumarate here. We see our FADH2 form. This would be another electron carrier. At this point, we've already formed two of our NADHs and 2 of our CO2s, a GTP directly, and now an FADH2. There's one more NADH that we're going to form in this citric acid cycle. So now we have succinate being turned into, being oxidized into fumarate. In reaction 7, fumarate is then turned into malate, and this is done with a hydration. So water's being added to the double bond here, and you guys would probably remember that reaction from the early days of our lectures. So fumarate is turned into malate. Fumarate turned into malate by the enzyme fumarase, and then finally in reaction 8, we're going to get malate to be oxidized into oxaloacetate. So fumarate into malate, malate into oxaloacetate. Now this regenerates oxaloacetate so that another round of acetyl-CoA can be added to it. We also get one more NADH. So to summarize, we've got all of these steps here. So the important parts are we added 2 carbons. We end up breathing out those carbons. We generate not 1, 2, but 3 NADHs. We generate an FADH2, and we get an equivalent of ATP. So this is the energy yielding steps. Now you'll notice we didn't have to put energy in anywhere into this. All of the energy that we used came from breaking apart high energy bonds. There was another high energy bond somewhere in here. Anyways, so that we can summarize as an equation. So acetyl-CoA with 3 NAD pluses, an FAD, and a GDP with some water will yield our coenzyme A, our 3 NADHs, our FADH2, and our GTP, as well as our CO2. So this would be sort of the net equation. Later, we're going to add up all of the ATPs that we've got, not just from glycolysis, but also for the citric acid cycle, and combine them with all of the ATP that we get from electron transport. How is this cycle regulated, you might be asking yourself? So similar to how glycolysis was regulated, the enzymes here are susceptible to allosteric regulation by either products of the later reactions or by substrates. So you can see here the citric acid cycle will actually increase, right, when low levels of ATP are present. So if ATP is, so I guess you could say, like how does this actually know when low levels are there? If low levels of ATP are around, that means you have high levels of ADP, right? Because ATP, when you hydrolyze it, it turns into ADP. So that would be an indicator. ADP would probably turn this on. ATP could act as an inhibitor, and so when there's less ATP around, it's no longer inhibiting it, right? And then, of course, the opposite is true. When you have high levels of ATP, you would want to be able to turn off these. So that makes sense that ATP itself, ATP might be an inhibitor, while ADP is the activator. And again, you can kind of see there are other molecules as well. NADH, it would make sense that if you had high levels of NADH already at the beginning of the citric acid cycle, then that means you don't need to keep doing the citric acid cycle. So that would inhibit. High levels of citrate, that should make sense also, right? Because if we're adding oxaloacetate and acetyl-CoA together, but there's already tons of citrate around, that should turn this enzyme off. We don't need to keep making more citrate. So there are 3 places where this is inhibited: the citrate synthase, the isocitrate dehydrogenase, and the alpha-ketoglutarate dehydrogenase. And you can see succinyl-CoA, which is the direct product of that enzyme, would be a inhibitor, just like citrate was here in this first one. ATP and NADH, of course, also activators and inhibitors. What's not mentioned here is that NAD plus might be an activator, right? Because if there's a lot of this around, you would want that to turn the process on. So that's regulation. Go ahead and see if you can fill in the answers here. The question is, how many of these are each produced during one turn of the citric acid cycle? So give it a pause, fill it out. So hopefully you said 2, 3, 1, and 1. Yeah, later we're going to have to, like I said, keep track of how many are produced during glycolysis as well, but that's for a later question. All right, let's look at electron transport. Electron transport involves carrier proteins and metal ions we'll see, and this all takes place in the mitochondria. So we're going to have to kind of switch the location here. What I'm showing you here is a cytochrome. Cytochrome carries electrons, and the way that it's able to do it is that the iron itself can be oxidized and reduced. In this case, the iron 3 plus will be reduced to iron 2 plus, meaning it gained an electron, and then it can go back to iron 3 plus when it gives that electron away. And so in a way, these guys are really good at receiving an electron and then giving an electron away. This is what an electron carrier does. It's held in place here with a heme group, similar to how our hemoglobin holds iron in place in our blood cells. Okay, So electron transport are coenzymes that are now reduced, are NADH and FADH2. They're going to enter into this electron transport chain, or as it's also known, respiratory chain. And these electrons are going to get passed from those NADH and FADH2s into different electron carriers. Now, each of these electron acceptors and electron carriers is going to have a job. Excuse me. The jobs that they're doing are not just moving the electrons across, but they're pumping hydrogens across a membrane. And that's going to be important. So we'll take a look at that. The energy of that membrane gradient of hydrogens that they're going to be establishing will be used to drive something called ATP synthase. And unfortunately, for whatever reason, I don't have a good picture of that, so I'll have to kind of jump around a little bit, but you'll see. Okay, so at the moment, if we keep track of how many NADHs and FADH2s have been generated so far, we know that we have 2 that were formed in glycolysis. There are 2 others that were involved during the oxidation of the 2 pyruvates into the acetyl-CoAs. And there's also 6 from the 2 turns of the citric acid cycle. So this is per glucose, right? Per glucose. There was the 2 NADHs as well, or FADH2s. So every glucose produces 10 NADHs and 2 FADH2s on top of the ATPs that are made directly. So we already have 4 ATPs. These guys have to go on to make more ATPs. The electron transport system, there's 5 complexes, and they're numbered. So here's complex 1, 2, 3, and for whatever reason, I don't have 4 and 5 shown in my slides, my later on slides, but this is 4 right here, and this is 5. 5 is ATP synthase. So we'll come back to this slide as we need to. Now, the electron carriers, there's also 2 electron carriers on top of the 5 protein complexes. Oops. There is cytochrome Q, coenzyme Q, and cytochrome C. And so these guys, similar to these proteins, are going to have some sort of metal that lets them be oxidized and reduced. These guys also attach to the inner membrane of our mitochondria. So let's take a second to kind of see where we are here. So we have, if we can imagine the mitochondria for a second, we sort of had this little situation. All right, so inside here is the matrix. That's this. So all of this stuff is in the matrix at the moment. Now, there's also a bilayer and an intermembrane space outside the matrix. So what's happening is there are proteins that are embedded in the matrix lining. That would be these guys. And they are going to pump hydrogens into that intermembrane space. Okay? That's going to increase the pH of that space, decrease the pH of that space, but it's going to build up a lot of hydrogen ions. Now, they're not going to be able to leak out outside of the mitochondria. They're going to need to come back into the matrix, but the only way to get back in the matrix is through protein complex number 5. And that's sort of like water through a water wheel, or like hydroelectric power, right? Water being forced to spin a turbine as it's moving from high pressure to low pressure. We get energy from that by making magnets spin around inside big coils of wire. Well, in the same sort of way, the hydrogens going through this molecule are going to mechanically cause it to start rotating. And as it rotates, these different little lobes here are all going to are like open and close and do different jobs. And essentially, they're going to catalyze this formation. They're going to be able to stick a phosphate on an ADP and make it into an ATP. It's like an ATP factory. Okay, so let's kind of start looking at them a little bit. But what's going to drive this is these guys are bringing hydrogens. These are where the hydrogens are essentially coming from, and they're also bringing electrons. Electrons are what are going to allow these complexes to move the hydrogens. So we're going to take a look at them one by one. So we'll start with complex 1. So this is where it starts. Complex 1 is where NADH transfers its electrons. So NADH, losing its hydrogen, is going to reform NAD+. So that's how this can go all the way back to glycolysis now and start, helping to oxidize more sugars. Okay. The hydrogen ions are also going to be pumped across the inner membrane into the to the inner membrane space, and then the electrons are given to this. They're given to this cytochrome. Cytochrome. Sorry, I keep saying that. They're given to coenzyme Q, and coenzyme Q, which isn't shown here. It's right here, but it would be, I guess, right next to it. I don't know why I moved this picture to show you guys like zoomed in ones, but coenzyme Q is here. And coenzyme Q will get reduced into coenzyme QH2. So it's holding those hydrogens for a moment, or there's other hydrogens around too, but it's got those electrons, and it is going to move between complex 1 and 2 to complex 3. So there's a little bit of stuff going on here. Let's take a look at complex 2 real quick. Let's take a look at complex 2 so that you can see what I'm talking about. Then we'll come back here to these. So complex 1 was right here. Complex 2 is this guy. So notice that NADH actually gives its electrons to complex 1, where FADH2 gives its electrons to complex 2. What this ends up meaning is that the amount of ATP that we get from FADH2 is not as much as what we get from NADH, and that's because we're putting the electrons in from NADH further upstream. They have more time to do more energy, right, before they get to complex 5, versus FADH2, which doesn't have that much. It doesn't have as much power, sort of say. It's like holding a ball, holding two balls up in the air, but one higher than the other, and then dropping them to see which one, hits the ground harder. The one that you held higher will hit the ground harder because it's got more time to build up, its momentum. Same thing here. The earlier you give electrons to this electron transport chain, the more work they can do. So FADH won't have as much ATP producing power as NADH. Now, the NADH gives it to coenzyme Q. It itself gets regenerated. Coenzyme Q then moves. And what we can see here is that for every 2 electrons that pass to coenzyme Q, 4 hydrogens get pumped across the membrane. So that's, our first building up of some hydrogens. Then we've got coenzyme Q also kind of moving back and forth between complex 1 and complex 2. So it moves here, gets reduced, it moves over here, moves down here, gets reduced, moves over here. And so it's just transferring electrons back and forth. I'm not actually sure what direction it moves in. It just moves in a couple directions, I suppose. Okay. So again, it becomes the same sort of reduced form, so I don't think it can be reduced at the same time by both of those electron carriers. So it must turn back into coenzyme Q before it can interact with this one, and then forms this, and then it turns back into this again before it can interact with NADH. But anyway, it moves back and forth between complex 1 and 2. It transfers these electrons to a series of iron-containing proteins called cytochromes. And so we're going to take a look at those cytochromes here in a second. Cytochrome C essentially is going to be the thing that moves between complex 3 and 4, but it's possibly giving its electrons directly to this iron sulfur protein. So let's take a look at that one. So complex 3 and cytochrome C work together. Complex 3 is the receiver of those electrons, and it looks like using the energy from those oxidations and reductions, it's able to pump more hydrogens across the membrane. So again, 4 hydrogens are pumped across for every electron that gets transferred. From coenzyme Q, the electrons get funneled to cytochrome C. And cytochrome C is going to be able to move to complex 4, which I don't have a picture of, so we'll have to jump back to my earlier picture. But again, more hydrogens are pumped across the membrane here in complex 3. Sorry, I'm scratching my beard. I hope that's not loud. All right. So in complex 4, 4 electrons from the 4 cytochrome Cs, so this is not a very good picture, it just shows 1, but there are 4 cytochrome Cs that are, there's lots of cytochrome Cs moving around, I guess. 4 electrons from 4 cytochrome Cs eventually get passed to this electron carrier. Electrons from here get combined with hydrogens and water molecules. So once the hydrogens come back through, they need somewhere to go. Oxygen is around, right? This is why we need oxygen for this whole process. The hydrogens and the oxygens and the electrons now are all allowed to meet and to react to form water. So water is the waste product here. All of these electron transfers, again, one more time, pump some hydrogens across the inner membrane space. And so now we have our built up hydrogen ions. And then the energy from these hydrogens are used to finally drive ATP synthase, which is complex 5. We'll talk about that one in just 1 second. The equation for this complex 4 step is 4 electrons, 4 hydrogen ions. I think one oxygen molecule makes 2 waters. So we get water as a result of the electrons and the hydrogens and the oxygens all meeting. All right. Complex 5. We call this oxidative phosphorylation, as opposed to substrate level phosphorylation, which we saw in glycolysis as well as in the citric acid cycle. Phosphorylation, ADP turning into ATP gets phosphorylated. So substrate level phosphorylation, forming ATP in just the cytosol. Oxidative phosphorylation is a result of this process, and it was called the chemoosmotic model. But it basically is a theory or a model that describes how energy how energy from the electron transport chain and that hydrogen concentration is used to drive the synthesis of ATP. And it basically says that complexes 1, 3, and 4 act as hydrogen pumps, and that because the hydrogen ions need to equilibrate and come back out, this is what drives that ATP synthesis. Now, in this chemoosmotic model, by passing through this 5th protein complex called ATP synthase, or complex 5, as I mentioned, it's able to drive this reaction, ADP into ATP. And so that's how complex 5 sort of fits into this. It is the site of ATP synthesis. I recommend you guys look at a YouTube video. I could probably post one or post a link to one, but there's a really cool modeled version of this whole process working and even showing ATP synthase like spinning and putting ATP molecules together. It's really neat. I recommend you watch it. So when NADH enters the electron transport complex or chain at complex 1, the energy of NADH can be used to form 2.5 ATPs, essentially. When FADH enters the complex, or at complex 2, it has enough to drive 1.5 ATPs. And so when we start to sort of calculate how many ATPs we can get, that's going to be the conversion factor, right? And NADH will yield about 2.5 ATPs, and FADH about 1.5. All right, how is this regulated? Similar to citric acid cycle, this is regulated by the availability of some of the reactants. So you have to have ADP to make this work. You have to have inorganic phosphate for this to work. You have to have oxygen. So having all of these in abundance will cause this process to go forward. Decreasing any of these is going to decrease how much ATP synthase or how much ATP is made. And this is like, when a cell is really active and ATP is consumed rapidly, then you'll have lots of ADP starting to build up, and that'll activate the synthesis of more ATP. And so it's kind of feedback regulation, just like our citric acid cycle. So too much of the products, too much ATP is going to slow this thing down. Okay. All right. So that is the electron transport system. Now, let's calculate ATP energy from glucose. Now, there's one other thing that we have to consider, is how do we get the, let's see, what's the word? I think it's the NADH that we're worrying about. NADH can't pass through the mitochondrial inner membrane. And so any NADHs that have been made in the cytosol, where glycolysis was, those NADHs have to actually get transported into the mitochondria where the site of electron transport is. And so there's a little trick that has to happen called the malate aspartate shuttle, and that's sort of what I'm alluding to here, but we'll get to it. We'll get to it in a second. So first off, ATP. The total ATP gets calculated by just combining all the ATPs together that were formed directly and all the ATPs that are formed from the electron carriers, not just in glycolysis, but also in the oxidation of pyruvate, the citric acid cycle, and the electron transport chain. So, so remember, glucose produces 7 ATPs, 5 ATPs from 2 NADH molecules, right? Because 2 times 2.5 is 5, and 2 ATPs directly. So there are 7 ATPs that are made just from glycolysis. There was an NADH times 2 made from the oxidation of pyruvate. Citric acid cycle made 3 NADHs, an FADH2, and a GTP, right? Times 2, because every cycle runs twice. And then the electron transport just turns all of these guys into energy. All right. But, as I mentioned, the NADH that was formed in glycolysis, it can't pass through to get into the mitochondria. So the hydrogen ions and the electrons from NADH can be moved into and out of the mitochondria by a transporter called the malate aspartate shuttle. And so what happens is malate dehydrogenase. So where's malate? Malate's right here. Malate dehydrogenase. Oh, I'm sorry. That's not malate. Malate dehydrogenase is right here. Malate dehydrogenase will actually catalyze the reaction of oxaloacetate and NADH to make malate and NAD+. So what happens is you've got an NADH floating around in the cytosol. You can't do anything with it. So turn it back into an NAD+ and let it go back to glycolysis. But using this oxaloacetate into malate conversion, malate is actually able to bind to a transporter and get carried into the mitochondria, into the matrix, where malate dehydrogenase in the matrix turns it back into oxaloacetate. So it just went in a big circle, right? But inside the matrix now, it took an NAD plus and made it into an NADH. So the equivalent of an NADH is now inside the mitochondria. And so that is how NADHs from glycolysis get moved in there so that they can be, added up. Now, oxaloacetate can get turned back into aspartate, which can be carried, which can go across the membrane. And that aspartate, once it goes across, can be turned back into oxaloacetate so that this whole process can continue. And again, that just makes sure that the NADH that was made in the cytosol can actually be utilized. So yeah, all right. Basically, it's saying everything I just said. I probably could have gone along with these slides. Okay, so ATP calculated from pyruvate, because remember we've got, well, I guess we're going to add it all up here in a second, but that was the glycolysis NADHs. Now we've got pyruvate. So in aerobic conditions, pyruvate we know can enter the mitochondria into the citric acid cycle. It gets oxidized into acetyl-CoA, carbon dioxide, and an NADH. So because there was 2 pyruvates, we have 2 NADHs, and those are going to account for 2.5 ATPs apiece. So 5 ATPs from the oxidation of pyruvate. From the citric acid cycle, I mentioned this already, we get a total of 6 NADHs from the 2 cycles, 2 FADH2s, and 2 GTPs, which are essentially 2 ATPs. So that would translate into 15 ATPs from the NADH. 2 FADHs produces 3 ATPs, sorry. There we go. And some direct. So we got to add all this stuff up now. So 2 turns of the citric acid cycle altogether produces about 20 ATPs. So we would say that, so we still haven't added everything together yet, but just from the citric acid cycle, from 2 turns, the 2 acetyl-CoAs turn into their carbon dioxides. So remember, that was 4 carbons, and this is 4 carbons, and 20 ATPs. So taking all of that together, we've got glycolysis, which generated 5 from the electron carrier and 2 directly, or 7. The oxidation of the pyruvate gave us 5 more. Citric acid cycle gives us 20 for the 2 turns. And so for 1 glucose, we can expect about 32 ATP. Now the numbers changed a little bit from when I was learning this stuff, 15 years ago. It's probably going to change again as we get better at sort of defining these things. But 32 about ATPs is what we can expect, 28 to 32 per glucose. So you need to know at which step these are generated, okay? Because on the test I could ask you for 1 cycle of the citric acid, 1 turn of the citric acid cycle, how many ATPs would we get? And you should know how to calculate that. But this table should be helpful. So here we go. The complete oxidation of glucose to carbon dioxide and water gives us a maximum of 32 ATPs. Now, we don't actually get all of the efficiency of energy that you might, from our food that you might think about in terms of ATP, but once I tell you where all that rest of the energy goes, it'll make sense. So what I mean is, what we can do is we can take glucose, kind of like a general chemistry experiment. We can put it in a calorimeter and see how much energy is given off by by combusting it, right? 1 mole of glucose should produce about 690 kilocalories. When we consider that 1 mole of glucose should generate about 32 moles of ATP, right? Because 1 glucose makes 32 ATPs, so 1 mole of glucose should make 32 moles of ATP, right? Kind of a ratio. 1 mole of glucose would make about 32 moles of ATP. We know that an ATP molecule has about 7.3 kilocalories in it. That only comes out to 230 kilocalories, which is not as much as what we know is the energy inside of glucose. So what's going on here? We can actually calculate what the efficiency is of our cells in converting the energy stored in glucose into ATP, and it seems that we can only convert about 33% of that stored energy into usable stored energy for our bodies. And you might think that that seems very inefficient. But I don't know if you knew this, on the coldest day of the year, standing in snow, completely naked, you could drink an ice cold soda and stay warm, or at least maintain your body temperature. And that's because all of that sugar that you're drinking in that soda, right? 66% of it is turning into heat. So heat is given off by these reactions, and that's where the majority of your guys' heat comes from, your internal combustion. You can generate some heat by, flexing your muscles and shivering and that kind of stuff. But the heat that your body is emanating is coming from this burning glucose inside you. And so that's actually a good thing, right? And if think about it, if we were able to convert 100% of sugar energy into ATP, then we would have to have some other system in our body that then took that ATP and used it for energy to make heat so that we'd be warm, right? I mean, there has to be warmth coming from somewhere. So that's where it comes from. It comes from the rest of the energy stored inside that glucose molecule. All right, I think that's the end. So this chapter focuses on the different reactions in the citric acid cycle, the different parts of the electron transport chain, the ultimate fate of those electron carriers and of oxygen and hydrogen and electrons, the fate of the carbons within our sugar that we ate, and then also how to calculate how much ATP from all of those different intermediates. All right, guys, so hope that wasn't too bad. Let me know if you have any questions or comments, and I'll see you in the next one. We got one more chapter after this.